Yes, good day, classy. Thank you very much. Today we are meeting to revise our Free State Province paper 2. As you can see, this is a preparatory exam that you can see displayed on our board here. Preparatory exam and grade 12 life sciences paper 2, September 2025. Please just take note that we are going to work the whole paper. We are going to work everything, and we will be also uh, getting into corresponding and related concepts of the question that we are going to answer right now. So without wasting any time, let's just go through it. I said it's Free State Province. We are going to check it very fast together right now. And as you can see, instructions, I said every time when you do exams, you always have to read the instructions. I'm not gonna go through them. I'm sure that grade 12, you are quite aware that instructions are very important. You follow them per question. So we are given here now question 1.1, which we are supposed to choose the correct answer. Multiple choice, I said section A class, section A, section A. We must get a total. Section A, we must get a total, which is 50 marks. Section A, we should get 50 marks, not less than that. Which one of the following characteristics applies to bipedal? We are saying bipedal. Remember, we talked about this also. If you refer on the channel, we are talking about organisms that are walking on two legs. Uh, it's a bipedal organism walking on two is what you call a bipedal, two legs. So now, which one of the following applies to bipedal organism? So we are to understand that a more backward position of the foreign magnum. There is no, it is not backward, it is forward position. When you look on the opening, that is the foreign magnum, it is on a more forward position, not backward. Backward, it is forward for quadruped. Backward position, it is for quadrupedal. And then a short pelvis and wider pelvis, a short and wider pelvis, which is what we actually talked about, that the bipedal has got a short and wide pelvis. I am sure that also go and check on the comparative analysis of a modern human being versus or the bipedal organisms against the other African apes. So now what happens is that I have a very good video that is explaining all this. Please go and check on that. And bipedal organism, he has a short and wide pelvis compared to the what? To this is quadra, long and narrow, which is also quadra, which is also quadra. And then we said for human being, it is what? It is the spine is S-shaped. For a bipedal is S-shaped. So what we are saying is the answer is what? The answer is B. So which of the following shows the process of DNA replication? DNA replication, we said making a copy of itself, making a copy of itself, which we say is an identical copy of itself. We call that what DNA replication. So now what happens is that what is the correct sequence of events? So if you look at it, we should understand that the double helix structure unwinds. That is true. Double strands and ZP complementary nucleotides pair up and two strands are formed. But you see, we are saying it unwinds, yes, double strands and ZP complementary nucleotides pair up, two, uh, two DNA strands are formed. Uh, we want to go through all the possible options that are there. So if you can see, we are saying the double strand helix structure, it unwinds the double strands unzip, complementary nucleotides pair up, and two DNA molecules are what are formed. So what is different here is that uh we don't form two strands. Two strands is where it is wrong, but we talk about two molecules, two DNA molecules. Two DNA molecules are formed, not strands. It is not strands that are formed, but we have got what? We have got molecules that are formed. So we can agree that when we have got question answer B is correct, that we have this is correct, double strands, it unzips, complementary nucleotides pairs up, and then two identical or two DNA molecules are formed, which is also correct. But if you see number C, it the double strands unzips, it also unwinds. This one starts, what starts first is unwinding, not unzipping. And then double strand structure unwinds, double strands unzip, one strand serves as a template, complementary nucleotides pair up, two identical, we are saying two identical double strands, it unzips, one strand serves as a template, and then complementary nucleotides pairs up, two molecules are formed. But then we cannot have two molecules formed when we have got one strand serves as a template. It does not, we don't have one strand serving as a template, but both of the strands are used to serve as templates. That is where it is wrong in terms of the strand. It is wrong there to say that the one strand serves as a template, but both strands, they serve as as templates in that regard. A trait that has a range of intermediate phenotype is an example of what? We talked about this variation. We are saying we talked about types of variation in this. Also refer to the channel. Types of variation, it is well presented. So when we have got a range of intermediate values, it means it has got a stretch, which you need to understand that we talk about continuous variation. Continuous variation, continuous variation, codominance is a form of expression, which is also form of dominance, which is not, we don't talk about that having a form of range of intermediate values. Discontinuous variation, it does not have a range. It has got no range. It is actually instant. And then complete dominance. So what we are saying is that if I'm to illustrate this, we say that this is a graph that can explain this is discontinuous variation, and then we have got what is called continuous, which is also having a stretch of values. This also shows what continuous variation, continuous continuous variation. So discontinuous, it has no set of intermediate values. Therefore, we can say that the answer is what is A. So, which one of the following is a possible genotype of parents that will result in a genotype ratio of 1:1 in F1 generation? We are saying P1, we are talking about parents one, the first generation of parents is the possible genotype of the parents P1 that will result in a genotype ratio of 1:1 F1, the first filial generation, the first filial generation. So when we take this one, if we take this one randomly, so I'm sure guys, I mean class, we are quite aware of what of genetic flow diagrams. This, if we can cross it, we are going to have B times B, which is going to give us what? We have got one gamete here, which forms the gamete forms, and then we have got B and B, which results in what? We are going to have a BB, which is 1. It is not 1:1. When we take this one, it is supposed to give us, we are going to have B, and then we also have B recessive across B, which is going to give us BB, and also we can also have BB, which is 1:1. 1:1. What is happening here is that we are saying we have got B crossed with what? With B. So we say that one and that one, they give us what? B, and then we take this one and this one, small b. So we have got heterozygous B versus recessive B, which will give us what? A 1:1 ratio. So our answer is what? Our answer is B, which is 1:1, 1:1. This one also, if we take it this, if we take this one, it will give us a single answer, which is going to give us 1, not 1:1. If we take this one, it can give us 3:1. This one can give us 3:1. I'm sure you know very well that a heterozygous complete dominance results in what? It results in a 3:1 generation, 3:1 ratio, which is equals to 75% as to 25%, which is what we are saying that is not correct. Our answer is 1:1, which is B, 50 as to 50, which is 1:1. Therefore, our answer is what? Is B. I'm sure that we are getting this very well. And then the following statements are sources of variation. Sources of variation, that is very correct to say, where do we get variation from? We have got variation from random fertilization, where cells are gametes, they fertilize each other randomly, crossing over, which we said that homologous chromosomes exchange genetic information during prophase 1. Random assortment of chromosomes also, which happens in metaphase 1. Mutations also, which is resulting in a change of the structure. But which of the following combination is a source of phenotypical variation? The phenotypical, we are talking about the physical appearance, the physical appearance of the organisms. Which of the following results in the physical appearance of the organisms? A combination of sources of variation. So what we are saying is that when we look at this one, random fertilization, it results in we are saying random fertilization, it results in what? In phenotypic appearance of organism, because the gametes are the ones which are responsible for that. Sorry, we are saying the gametes are responsible for fertilization. So gametes random fertilization, we can look at it and also need to understand that the correct answer must have we are saying the correct answer must have combinations that actually expresses the meiosis itself is just an arranging process, so that actually change how it actually looks. So our answer must be 1, 2, and 1, 2, and 4. 1, 2, and 4. 1, 2, and 4. We are saying 1 is correct, random fertilization, crossing over, and then also mutation. So our answer must be 1, 2, and 4. So which is 1, 2, and 4, which is what? Which is B. The results in what? The results in a combination of phenotypical variation. This question comes back again. Evolution represents below the pelvises and skulls of hominids. The pelvis and skulls of hominids that are given, question number 6. So we are given, this is, we are seeing that it is narrow and long. It is wider and also what? And short. I'm sure that if you also refer on our channel class, we have got these well explained. We have got an explanation that covers these what to call structural similarities and differences where the diagrams are even attached. We are saying this is a backward position of the foreigner magnum, which is more backward. Backward position, we said that the backward position indicates that there is a what? This is a quadruped. This is a quadruped. You can see also when we look at this number 2, you can see that it's a what? It's at a more forward position, which results in a vertical sitting of the what? Of the foreign magnum. So therefore, we can also see this one also is at a backward position. Backward position, what does it mean again? It means quadruped. It means quadruped. So let's look at the question that are there. The correct combination of pelvises and skulls is, the question is saying represent the pelvises and skulls of hominids. So we are given these, and then we are supposed to understand to say the correct combination of pelvises and skulls is what. So which skull matches the pelvis, the skull, the pelvis? We want to see that they match each other to correctly represent the diagram that we want to understand. So we are saying pelvis A, skull 2, pelvis A, it has got skull 1, skull 2, and we look at pelvis 3. So what we are saying is pelvis A, pelvis A, pelvis A, it must represent a skull number what? Pelvis A must represent a skull number, pelvis A must represent a skull number 3. In that regard, therefore, it cannot be pelvis 1 and skull number 1. Therefore, A is wrong. And then, so what we are saying is pelvis A must represent skull number 3, which would always just mean to match that skull number 3. It must match pelvis A, which is representing our evolutionary hominids. So now, what we need to understand is that we have got skull pelvis B represents number 2, pelvis B represents number 2. Number 2, which is, of course, we said that it is a hominid, it is a human being, which has to show a forward position and also a vertical or a bipedal structure. So that is also to say number 2 is correct, and pelvis 3 represent skull number 1. Pelvis 3 represents skull number 1. So what we need to look at is the similarities or the structural appearances of these hominids in terms of their what? In terms of their position, in terms of their evolutionary changes, to say pelvis. If you can check, for example, one other thing that makes you to understand these answers is that you can strictly match number 2, at least if you have got a specific pelvis and skull that you know, like we are saying, pelvis A must represent skull number 3, and also pelvis B must represent skull number 2, because we are talking about the shorter or wider and short pelvis represents a bipedal organism which has got a vertical or it has got a more forward position foreign magnum. So what we are saying is the correct answer must represent a combination of 3, 2, and 1 according to the pelvises. Pelvis A, B, C. Pelvis A represents skull number 3, and pelvis number B represents skull number 2. Pelvis B represents skull number 2. So we are saying, according to say to this, we are saying this and that they match. And then also A, if we're gonna put it differently, we are saying A also, it matches with what? With 3. And then number C, it matches with what? Number C which we can also put a different color to say C, it matches with what? With A. That is the relationship that we are talking about in these pelvis. So the same color represents those which are matching. Blue is matching to blue, green is to green, and black to black in regard to these pelvises and what is being asked in this respect. Uh, which of the following statements relates to Lamarck's theory of evolution? Lamarck's theory of evolution, the alleles of favorable characteristics is inherited. We are saying favorable characteristics are inherited. Uh, this was also according to Charles Darwin's theory. And then acquired characteristics are inherited is the major principle of of Lamarck's theory. Lamarck's theory has got his core principle, which we talk about acquired characteristics. Acquired characteristics are inherited. Acquired characteristics, we said characteristics that are or obtained by an organism during its lifetime are inherited, which made the Lamarck's theory to be what? To be discarded or not to be considered. So the correct answer is B. Acquired characteristics are inherited. And then we have got question number 8 to karyotype, the structure and arrangement. We are saying the karyotype, we look at the structure, we look at the arrangement, we look at the shape. It is the structure, it is the arrangement, and also the shape, and the shape of what? Of our, it is also the arrangement, the structure, and the shape of what? Of the chromosomes. So now, what we need to look at is that we are given the karyotype that we are given here. So the chromosomal composition of this cell is dash. The chromosomal composition, we are saying how many chromosomes are in this cell. So as you can see, what we need to understand is that the chromosomes, we are just counting the total chromosomes that are within this cell, all of them from number 1, 2, 3, our autosomes, which of course we find out that on chromosome number 16, we have got triplets. We have got triplets on chromosome number 16, and then our gonosomes, our gonosome, chromosome 23, our sex chromosome, it is actually showing that they are also what? We just have two, which represents a female anyway, because we have got XX that are given there. So now, what we need to look at is that what does the question wants us? The question is saying the chromosomal composition in this cell is dash. How many chromosomes do we have? We have got, instead, we are saying a normal cell, a normal human cell have got 46, where if all of them are double, but in this case, we have got chromosome 16, which is having a triplet. So the triplet means that we have got 47. We must have 47 chromosomes. We have got an extra chromosome, which makes them what? 47. So number 80, it is what? 47. 47 chromosomes that are in terms of this question. We have got 47 chromosomes that are in there. And then how many over carry the X chromosome if meiosis occurred in the above cell? How many over carry the X chromosome? Number 9, how many over carries how many over would carry the X chromosome if meiosis occurred in the above cell? If meiosis occurs in the above cell, how many of them can carry the what? How many of them uh, can carry the X chromosome? So what you need to understand is that an X female undergoes meiosis and donates an X to every single ovum. So 4 out of 4 that can be formed in that case. So we are saying an XX female, XX represents, I'm sure I remember that XX represents what? Female. It represents female. So what we are saying, or what you need to understand is that when it undergoes meiosis, if it undergoes meiosis and donates an X to every single ovum, 4 out of 4 of them can be formed. So it is four, which can also, X, four of them can also carry the X chromosome if meiosis takes place there. And then we are given a filial a filial atrial fibrillation is a condition in which the individuals have got an altered heart rhythm, the pumping, the heartbeat. The condition is an inherited condition that follows an autosomal dominant inheritance pattern. An autosomal dominant inheritance. So we are saying it is caused by what? Dominant inheritance pattern by a dominant allele. And then shaded individuals in the following pedigree have this condition. Naturally, we know that when we see a pedigree, a shaded one represents what? It represents an affected person. And then, of course, we have got confusions or sometimes illustrations which might represent that a person can be what? Can be a carrier, but therefore we are saying it is unshaded. It is unshaded. Therefore, what it represents when the person gives rise to a child who is affected. The mother is what is a carrier. We are having an affected son. So affected son means that the mother is unshaded. If the mother is unaffected, therefore it means that is a what? Is a carrier. That is one thing that we need to understand, that whenever we have got in unshaded uh unshaded circle which represents an affected mother, but we have got an affected son. When we have got an affected son, what is the outcome or what is the meaning of that phenotypic expression of that mother? She is what? She is a carrier because the mother gave a recessive allele to this son here. So now, by studying the pedigree diagram, it could be concluded that it can be concluded what? That autosomal dominant pedigree. One thing that we need to understand is that autosomal pedigree, dominant pedigree, affected individual come up every generation, and why? And generation individual one must carry one affected allele and one normal allele. So therefore, what happens is that in this person, we are saying the person must have a heterozygous condition. So in this case, we are saying uh, the person, by studying this pedigree, we can say generation Y, individual one is heterozygous. Generation Y, which is given here, generation Y is a generation here because we are looking from it from here up to there, which is a generation who are children who are born from generation X. So what we are saying is that autosomal dominant pedigree, what happens is that an affected individual comes up every generation. An affected individual comes up every generation, and therefore and generation every generation and generation Y, individual one must carry one affected or one rogue allele and one normal allele, which actually represents what? A heterozygous condition. So in other words, what we are saying is that our conclusion is saying that question is the answer is what is Y. Generation Y, conclude that generation Y individual one is heterozygous. Generation Y individual one is heterozygous. That's what we need to understand. Let's do the question number next. Biological terms are the disorder, genetic disorder. Uh, Genetic disorders are controlled by recessive alleles that are found on the gonosomes. We are saying that are found on the sex chromosomes. They are found on the sex chromosomes. So now, what are we saying? What is the condition? These are called sex-linked disorders. They are called sex-linked disorders. They are called sex-linked disorders because we are saying the conditions are caused by a recessive allele that is carried on an X or on which is carried by by a sex chromosome, which can be which is recessive, class. So it is called sex-linked disorders or sex-linked diseases. A testable statement that can be supported by scientific evidence. We call that a hypothesis. Hypothesis, if you look up in our hypothesis, it's a statement that can be supported by scientific evidence. So when we are doing a research, when a research is being done, we have got an educated guess that is given, which is actually premised or which is set up in order to control and derive the research. So when research is done, we are finding scientific evidence to either support or dispute the statement. So a testable statement that is supported by scientific evidence is actually a hypothesis that you need to know because it is supported by the research findings, which are actually facts based on scientific evidence. And then a genetic cross involving one characteristic, we say that one means what? Means mono. So it is called a monohybrid. That is called monohybrid. I'm sure you still remember that mono means one, and then we said if there are more than that, we look at the opposite of mono is what? Dihybrid crosses. You remember, we did the dihybrid crosses again. Just look also on the channel. You shall find out that these are also clearly given there. And then the phase in a cell cycle during which DNA replication occurs. DNA replication, it is also a question that is always repeating itself. I once answered it also I once answered it when we in another section of questions. So what you need to understand is that DNA replication it occurs or clocks during the S phase of interphase, where S is actually for the synthesis process. So we say that it occurs during the S phase. It occurs during the S phase of the interphase. S phase of the interphase. I'm sure that we remember again when I did show this process of DNA replication, the cell cycle. There is a video also that you can refer to, which is quite just interesting and also interrogative. The dominance of where the heterozygous phenotype is an intermediate between the two dominant homozygous phenotypes. When we say these are the dominance where the heterozygous phenotype is an intermediate, heterozygous, I'm sure you remember that we talked about these expressions, genetic terminologies, to say heterozygous is where we have got a dominant and recessive allele. A heterozygous phenotype is an intermediate. It is an intermediate between the two dominant homozygous phenotypes. So what happens is that it is shown by the dominant alleles. So what we call that, it is called incomplete dominance. It's called what? Incomplete dominance. It is called incomplete dominance. That is called incomplete. That is called incomplete dominance. And then the phase during meiosis, nuclear when membranes will reappear in two daughter cells. It is telophase what? It is telophase 2. I'm sure that we can remember that the phase where the nuclear membrane rewrap the chromosomes like a cling film is actually what called the telophase 2. Telophase, it's called telophase 2. A change in an organism's nucleotide sequence of the DNA molecule. We call that what? Mutation. Mutation. Change in a nucleotide sequence of a DNA molecule. We call that mutation. That is called mutation. And the genetic condition is characterized by the absence of blood clotting factor in humans. We call that, I'm sure I remember we've been talking about the homophilic condition, which also we normally refer, that is quite common when we talk about it, also referring from what? From the Bible, the woman who has got a flow of blood. So we say that it's called the homophilia. Homophilia. It's called homophilia, where the person cannot stop bleeding, or it takes long to stop bleeding. It's because the person has got absence of blood clotting factor. We call that homophilia. And the start of geological distribution of species providing evidence of evolution. Sources of evidence of evolution. Also, please check on the channel, class. Check on the channel. We have got a video presentation that talks about the article sources of evidence for evolution. So we call that what? We call that biogeography. Bio means life. Biogeography. I have got a very nice, interesting video which explains biogeography, which explains comparative anatomy, which explains the cultural evidence, all that. So please just also look on it, check on that. Evidences of evolution, sources of evidence for evolution is well explained. The theory of evolution proposed that the offsprings who inherit the alleles of favorable characteristics. We say that that is a theory of evolution by natural selection. Evolution by natural selection. Please check on that again. These are just interesting chapters which we have taken time to work on them. So please just keep on connected. We need to look at question 1.3. I'm sure you know that we say A, both none, or B or A alone. So we are given these questions here now. Cultural evidence to support human evolution. Cultural. So art represents culture. Art, it can be done to represent culture. We are saying we have got the life of people being expressed through arts and culture. So we have got tools again. The tools also are a cultural evidence. So what you need to understand is that this question 1.3, it's both A and what? And B. Is both A and B. A and B are both correct. They are both sources of evidence to support human evolution, cultural evidence that can support human evolution. We are saying that is what both A and B. So stem cells, when we look at stem cells, what you need to understand again is that we talk about biotechnology, we talk about all these. So stem cells, they actually are undifferentiated somatic cells. Stem cells are undifferentiated somatic cells, which makes the answer to be A only. It is going to be A only. Multiple alleles. We are saying which is controlled by more than one allele or more than two. So what we are saying is that when we look at this one, multiple alleles, they part into what? Into blood groups. If you look in blood groups, we have got many alleles that controls. We are saying we have got blood groups is controlled by three alleles. It controlled by three alleles. Homophilia, if we look at it, it is actually controlled by two. It is actually controlled by two. So if we look to say that which one is having multiple alleles, it is controlled by only two alleles, which are carried by what? Which are carried on the X chromosome. So now, what happens is that uh, the answer must be what? Must be A only. Blood groups is controlled by multiple alleles. Is controlled is an example of multiple alleles. And then we have got the diagram representing protein synthesis. We say that protein synthesis is what we call protein formation. Protein synthesis is what we call protein formation. So now, what we need to understand is that this is also on the channel class. I have also a very good presentation which is explaining the process of what protein, the process of protein formation. It is a quite comprehensive video that explains the process of protein formation. So the first question is saying we are given this, we can say that this is a chromatin network which is in the nucleus of a cell, which is in the nucleus of a cell. So now what we need to answer is that we have got multiple questions there. We can say these, we can say tRNA, transfer RNA, N, we can say that N can represent our mRNA, the single strand that we are seeing that goes through the ribosome. And then we have got our protein chain, which we say we have got our what? Peptide bond. So now, what we can say again is that these we can call them what? I want you to understand that these we can call them the three nucleotide bases on mRNA. We say they are called what? They are called codons. We call them codons. We have got our codons there, and then we have got there, we call these anticodons, which is also three nucleotide bases that are found on tRNA. We call these anticodons. Anticodons. You understand? So, having said all this, I'm sure you are going to see and you are going to make it easier when it comes to, of course, we say all these are what we call these are called amino acids, which are brought by tRNA. You understand? So if we can go into this, identify structure K. Structure K, if you look on your diagram, what is structure A? Structure K. Structure K can represent, it actually represents the cell nucleus. I have already mentioned that K, structure K, it is the what? K is circling, is that one, which represents the what? It represents the nucleus. K represents the nucleus. That is what? This is K. And then molecule Q, our molecule Q, uh, molecule Q, it actually represents the peptide bond. It actually represents the peptide bond. Peptide bond. A chain of we are saying the peptide bond which is formed through the ribosome as the mRNA that is passing through is being read by the what? By the ribosome there. So we have got our chain which is forming a peptide bond, a chain of amino acids forming the peptide bond that will form what? Proteins. And then give the letter of the organelle where translation took place. We are saying a position where translation occurs. It occurs by what? by letter. It actually occurs where organ is taking place. It occurs at the ribosome. Like I said, the ribosome is represented by what? By O. O represent the ribosomes where translation is occurring. And then a tangled network of DNA. The tangled network of DNA, it is found in the cell nucleus. So this can be what? This is L. It's what? DNA. This L represents the tangled DNA network of DNA. We are saying it is L, which is found in the cell nucleus class. That is L. And then hairpin-shaped nucleic acid. A hairpin-shaped nucleic acid, we need to understand that is what? That is where we have got our mRNA, I mean our tRNA. tRNA is the one that looks like the shape of a hairpin, a hair-shaped, hairpin-shaped nucleic acid. So our tRNA, we can put that is M. We can see that this is M. It represents our tRNA, like I had already mentioned, the tRNA transfer RNA. So our pin-shaped nucleic acid is what? Is M. These are the answers that you should take note of. And then determine the complementary DNA-based triplet for codon P. The complementary DNA, the complementary DNA. Remember that we are looking at DNA-based triplet for codon P. For codon P, let's just see where P, codon P, the complementary DNA-based triplet. We are given that that is P is equals to. We are given that P is equals to the is equals to AAU. P is given as AAU, which we are seeing there on what? On the diagram. P, we are saying is equals to AAU. This is what? mRNA. mRNA. So in terms of the DNA, what is the complementary? What is the complementary base triplet? So in this case, what you need to understand is that this we are saying this is the what? This is the codon. This is the codon. So what we want is that we are saying give the complementary base triplet. So you should understand to say in DNA, which are the complementary bases that pairs to what? To this codon that we are given. A, it pairs with T. A pairs with T. A pairs with T. A pairs with T, and U, as you can see, we are saying U, it is representing T in other ways, which must pair up with what? Which must pair up with A. So the complementary base triplet in DNA is going to be TTA, given that our codon is this one, which is AAU. So it is going to be TTA according to the required or the corresponding complementary pairing of bases that we understand. And then name the amino acids from left to right as they will bind on molecule N. We are saying from left to right. We are naming them from left to right as they bind from left to right as they will bind on molecule N. Molecule N, we are given that molecule N, we are given our molecule N, we are given that we have got the codon sequence, which is AAU and GUC. I'm saying we are looking at molecule N, we are looking at this and that, and then we are going to read them from left to right. So we have got AAU, that is question 1.4.4. Question 1.4.4. So what we need to understand is that the amino acids from left to right as they will bind on this on molecule N. So we are to understand class that when you look at this, we say that we have got A, U, which is also when also we are saying AAU, which is given in this is AAU that I'm talking about. AAU, I'm talking about this AAU, and we have got GUC, AAU, and we also have the second one, GUC, AAU, and we also have GUC. This is our molecule N that we are talking about. This is our molecule N that we are referring to. So now read it so that amino acids that can bind on it from left to right. So we need to look at the corresponding amino acids that can bind to this one, AAU, according to our amino acids, uh, what is it, molecule table. You understand that AAU, it represents, AAU, it represents, uh, it is known that it represents aspar asparagine, and then uh, GUC, it always represents villine. So now, what we are saying is that when you read it from left to right, they want you to give these as the answers to say, we start with this is 1 and this is 2. So you take them from left to right, which is going to say that this is asparagine, and then we have got what? We have got feline as the second one. I hope we are getting this very clear. We are getting this as much as possible. That is what we are looking at. That is what we are looking at. Molecule N there, and then we look at it AAU and GUC. I hope we are getting this very well. So let's just go to the next question. The next question, it is showing complete the table. Complete the following table representing famous scientists, the hominin fossils they discovered and fossil site where the discoveries were made. So we write only the answer to the question number. So question 1.5, what is happening is this is one of the most outstanding concepts that you need also to understand class, that when we go to exams, you need to be able to understand, and you should be able to relate and get this very well, that what happens is which scientists discovered certain fossil, the name of the fossil and where it was discovered. So therefore, it is very important for us to go to exam understanding those. So 1.5, we are saying misplace, misplace, what scientists responsible for the discovery of misplace. I'm sure that you remember that misplace was discovered by Robert Broom. This was by Robert Broom. Robert Broom discovered misplace. Misplace was discovered by Robert Bloom, which you remember that it was nearly a complete skull of the species, which was called the name of the species was called Australopithecus, which was actually the Australopithecus africanus. Australopithecus africanus. That is 1.5.2 Australopithecus africanus. I'm sure that this was actually in Fontaine caves that you can know, which was actually significant in major excavation and effort in the cradle of humankind, and also the World Heritage. Was actually like in the somewhere like 40 kilometers northwest of Johannesburg. That is where misplaced was discovered. And then question Raymond Doty, the name of the fossil that was discovered by Raymond Doty, it was also, we say that Roman date. If you look at that again, you discover again that he discovered the Taung child. Taung child was discovered, which was also Australopithecus africanus at the fossil site. It was Taung, and also northwest. The uh, the discoveries that were done that were in this regard. The Tawing child, I'm sure that you remember again that this was also uh, discovered by Deti, which was somewhere in 1924. And then what we need to understand again is that it is one of these uh, historical heritage sites in anthropological fossils of the 20th century, which actually provide the evidence of the early humankind on upright two-legged walking position. And then we also talk about the brain size, which was around 400 to 500 cubic centimeters, which can also be comparable to a modern adult chimpanzee. And then we have got Karabo, Karab 1.4, 1.5.4. We say this was discovered by Matthew Baker. Matthew Baker discovered uh, Matthew Matthew Baker Matthew Baker discovered Karabo, and then it was Australopithecus sediba. That was Australopithecus sediba, the Karabo, Australopithecus sediba. So now, we actually want to understand that this was discovered by Matthew Baker. We should not forget that Matthew Baker discovered Karabo. And then one other thing that you need to know is that this one, it actually means the Southern Ape, which is the Southern Ape, which actually is believed to be a juvenile male that was around 9 to 13 years old when that boy died and was found alongside the remains of an adult female, which also showed that there was he had his mother there. We've got question 2.1, which is actually showing the undersea earthquake triggered a devastating tsunami, claiming the lives of at least 225,000 people across multiple countries due to overwhelming number of casualties and severe damage to infrastructure. Mass graves became necessary to bury these bodies. Talking about the ease in burying, DNA profiling was used to identify some of the victims. Months after these bodies were buried, DNA samples were taken from the bodies before burial to compile the DNA profile, and were they also compiled from the samples provided by family members. These samples were collected from toothbrushes, hairbrushes, clothing, and of the deceased. This process helped to identify some of the victims. You understand? So we are talking about this one. Also refer to channel. There is also a good presentation that is also DNA profiling. DNA profiling. It actually, there is also a very good presentation about DNA profiling on the channel. Please go and check on that. Please go and check on it. It is quite interesting, and I'm sure that you enjoy enough there. So question first is saying what tissue could be collected from the toothbrush to use as a source of DNA, as a source of the DNA. when DNA profiling with other sources, there was a first question that the previous other question that we answered is sources of evidence of what? Of DNA profiling. So we can find the sources as all these ones: saliva, hair, clothing, and so forth and so forth. So now, what are those ones that can be found there? We can talk about buccal epithelium cells, buccal epithelial cells, which are found. We talk about these as the cells that are found on the toothbrush, epithelial cells. We can also talk about cheek lining cells. Cheek is like cheek lining cells. Cheek lining cells, they can also be used. They can also be found on these toothbrushes. Explain how DNA profiling was used to identify some of the tsunami victims. Question 2.1, 2.1.2.1.2.1.2. How DNA profiling was used to identify some of the tsunami victims. 2.1.2. Let me just write it. 2.1.2. How DNA profiling are used to identify tsunami victims. Tsunami victims. So what we need to understand is that when we look at this, we are supposed to understand that we need to look at this and then understand these things to do with the process of DNA profiling. So DNA profiling, we need to look at the scientists. One, we are saying scientists, the scientists first, what they do, they extracted DNA from the victims. They extracted DNA from victims. They extracted DNA from victims and also magnified the short tandem repeats. That I'm sure that you remember that these STR regions, STR region are using which they were using the polymerase chain reaction. Polymerase chain using the polymerase chain reaction, PCR. So we are saying again, in this case, what we are saying is that they separated them via a and then separated them. They separated them through what is called we have got what is called gel electrophoresis. Gel electrophoresis. Actually, so therefore to create barcodes. To create barcodes. To create barcodes. So therefore, these barcodes are like barcodes which are like fingerprints. Like fingerprints. Like fingerprints. Like fingerprints from the relative's finger, from the relatives as toothbrush, from the relatives toothbrushes or hair brushes. or hair brushes, so that so therefore what we are saying is that matching barcodes, where they find that by identifying matching barcodes, by identifying matching barcodes, by identifying matching barcodes, I mean matching bands. In other words, let's say matching bands, matching bands. By identifying matching bands, therefore they give positive identification. They gives positive. They give positive identification. They give positive identification, which we are understanding that bringing closure to families. Positive identification, and therefore closure to the families, and therefore closure to families. They also bring closure to families, which we say that this was after the tragedy. You understand? So what you need to understand is that if you look at this explanation class, we are saying how the DNA profiling was used to identify these persons. So what you need to understand is that DNA is extracted by scientists from what? From the victims. The victims, DNA is extracted from the victims by scientists, and they magnified or they enlarged the what? The STR, which I say that you need to understand that the short tandem repeats, which is actually specific regions in DNA where a pattern of two or more nucleotides is repeated. So in other ways, they actually vary they can actually vary between individuals to individuals, which therefore can be used to identify what people, and also using the polymerase chain reaction. The polymerase chain reaction that we are talking about, I'm sure that you remember that this is also is a rapid also. It is just uh, a rapid and fast analysis in terms of the DNA, which actually it is done through the laboratory technique, where it is used to what? To amplify specific regions of DNA so that small and degraded DNA samples can be analyzed. So what happens is that it actually also we are saying degraded and small DNA samples to be what? To be analyzed, because we are using a laboratory technique there, which actually amplifies the specific regions of what? Of DNA, in order to identify those degraded or small samples of DNA. And then we are saying they are separated through the geoelectrophoresis to create barcodes like the fingerprints from the relatives, and these from relatives to the brushes, hair brushes, and then by identifying matching bands, this gives a positive identification and therefore closure to families after the tragedy. That is one thing that you need to understand in this respect. So explain your answer. Okay. In some cases, no personal in some cases no personal effects of the victims could be found. No personal effects of the victim could be found. Which family member, the victim's spouse or the victim's parents, could be used to provide the sample for comparison. In this case, we use the parents, victim's parents. Victim's parents is used. Victim's parents is the one who has got a possible source of DNA sample that can be used in this case. So without wasting time, we are saying the victim's parents is the one that is used. The victim's parents. So now explain your answer. What we need to understand is that parents actually they donate half of parents basically each donate half of their genome. So what we need to understand is that so ben sharing is high in terms of the parents than in what than in the spouse as what than in the spouse. So what we are saying is that the reason here is that there is because there is because there is high bench sharing from the parents than spouse than spouse. What we need to understand is that because of parents, they share their genome to their child. Parents share half. They share half of their genome, half of their genome to their children. I remember that the genome we are talking about the total genes that are in an organism. So during reproduction, parents they do share half of they share they actually share uh, half of the genome of their genomes to their parents. I mean to their children, unlike the spouse shares zero DNA with the what? With the victim. So the victim's DNA and the spouse's DNA cannot match. So if we can actually look at the victim's STR profile, we can find that parents have got the best relative or they've got the best similar DNA profile to the victim than the spouse. They are unrelated parents, I mean, the victim and the spouse are totally not related, so their DNA cannot match. So the last question is saying, describe the structure of a DNA molecule. The structure of a DNA molecule, the structure of DNA molecule. This is also, please refer to channel also. You must also refer to channel class. Refer to channel. These are last year videos that we have the structure of DNA. So what we need to know is that the DNA is a double helix. DNA is a double helix. DNA is a double helix. Actually, we need to understand that DNA is a double helix with strands that are twisted. That are twisted. Uh, that are twisted along each other. That is one other thing is made up. DNA is made up of nucleotides. It is made up of nucleotides that is nitrogen bases, which is the nitrogen basis. We say there is adenine, there is thymine, there is guanine, and there is cytosine. We have got also phosphate group, and what? And deoxyribose sugar. And deoxyribose sugar So what happens again is that DNA complementary bases are held together by weak hydrogen bonds, by weak hydrogen is bound together by weak hydrogen bonds, which you should understand that A pairs with thymine and C pairs with what? With guanine. So the number of nitrogen, the number of each base pairs is equal to each other, which we say that A is equals to T and C is equals to what? To G. So basically this is along which what you should understand that they are the structure of DNA structure, describe the structure of DNA. That is what you can actually have to put across as you go through this. Are we together there? I hope we are, I hope we are understanding and we are looking at it, getting it very well with regards to these questions. So now, if you go on again, we go to the next question. The next question, it wants us to look at, so we are going to the next question, which is showing the diagram that represents the phase of meiosis. A diagram that represents the phase of meiosis. So now, what we need to look at is that this is the structure here, which we are given, a stage of meiosis that we are given in testes. In testes, so if we are saying testes, this is in males. This is also spermatogenesis. If we are going to just quickly take note of it, it is spermatogenesis that is taking place here. So now, what is the question saying? Identify the phase. Identify the phase of identify the phase represented in the diagram. If we look at the phase that is represented on the diagram, we can actually relate and say that the phase that is being shown here, it is what? If we can look at it, we can actually relate and see that the phase that is represented on the diagram, we can see that this is anaphase. This is anaphase 2. The phase is anaphase 2. I'm saying also, if you look on the channel again, class, we have got nice extensive explanations of the cell cycle, of meiosis process. State 1 function of A, state 1 function of A, this is the spindle fibers. This is the spindle fiber. So therefore, what is the function of the spindle fiber? The spindle fiber, what is the function there? It's actually, we need to understand that it is actually the spindle fiber contracts to pull chromatids apart and also control the position of chromosomes during the cell cycle. We are saying contracts to pull chromatids apart. In other words, what happens is that the spindle fiber, it is there to control or to influence the positioning and the characteristics of a cell by positioning the chromosomes or chromatids. The positioning of the chromatids and chromosomes is controlled by the spindle fibers. And then in this phase, give the number of centrols. In this phase, give the number of centrols. We are saying the central mass that we have, we are given that give the number of centrols. We are saying we actually have 2. We actually have 2. So what is the centrols? Give the number of centrols. We are saying we have got what? We have 2. And then there's the number of chromosomes present at the beginning of the of this meiosis. The number of chromosomes that are at the beginning we know that at the beginning of the cell, this is a human cell, a normal human cell, it's got 46 chromosomes. It's 46, and then gamete that will form at the end of meiosis, the number of gametes that will form at the end of meiosis, we say again, we would have 4, which I'm saying that you remember this has been, we also actually explained that meiosis, it wraps up with the 4 cells. We have talked about this. I have explained it also in the previous question, and then describe the behavior of the chromosomes during, describe the behavior of the chromosomes during the phase in the above 2.4, 2.2.4. What we need to understand is that the behavior is that chromatids, they actually synchronously split at the centromere. We are saying one thing is that sister chromatids, they together split, which is they together split, they the chromatids, they do synchronously, they split synchronously splitting at the centromere. They split at the centromere. What again? Which actually then migrate centromere first towards the opposite poles, and then migrate first the centromere. They migrate first the centromere. The centromere actually they migrate towards, or they migrate towards opposite poles. Migrating towards the opposite poles. They migrate towards opposite poles while the spindle fibers shorten. The spindle fibers, they shorten class, while the spindle fibers, they do shorten. While the spindle fibers, they shorten. So now, explain Mendel's law of segregation during meiosis. This I also say it has been explained on the channel again. Explained on channel also. Mendel's law of segregation, it is also explained on the channel. So what we need to understand is that this question, what we need to understand is that question number 2.4 and 2.2.5, 2.2.5. What we need to understand, class, is that each pair of alleles separate during gamete formation. Each pair of alleles they separate during gamete formation. They separate during gamete formation. Each pair of alleles separate during gamete formation, and then which we are saying that each pair of alleles are separate during gamete formation. They actually separate from each other, and then each gamete receives one allele, and each gamete receives one allele. Each gamete receives one allele. each gamete, what happens is that when that occurs, each gamete receives receives one allele, which is one allele from each pair. They receive a pair each formation. We are saying each gamete receives one allele from each pair, and then random assortment resulting. What happens is resulting in a mixture, resulting in a mixture of different alleles combination, resulting in a mixture of different alleles combination. That is basically what we talk about the random assortment, or what you call when we're looking at the law of segregation from Mandel. It talks about pairs of chromosomes, pairs of alleles are separating. Each gamete receives one allele, and then we talk about resulting in what? In a mixture of different alleles combination in an offspring. That's what we are saying, or that's what we need to take note of. 2.3, 2.3, we are given this 2.3, we are given this question there, which we need to look at. Henry VIII was King of England from 1509 to 1547. And then what we need to look at is that he was the king from then. During his reign, he actually had 6 wives. Infamously, he sent 2 of his wives, Anne and Catherine Howard, to their death on the executioner's block at the Tower of London. One of the reasons he had these wives executed was the fact that they had not given birth to a male wife. There was no a male heir that was given birth by these guys. So now the question is saying, which parent determined the sex of a child? Which parent, the question is saying, which parent determines the sex of a child? Which parent determines the sex of a child? Which parent determines the sex of a child? So I'm sure that we have also a question that I presented to you guys, where we talk about the male, child sex determination, where we talked about sex determination in children. Which parent determines the sex of a child? So now, the sex of a child is represented by what we say that the father determines, or the male parent determines the sex of a child. Father, or we can say in this case, Henry VIII, according to this question, is the one who determines the sex of the child. Draw a label diagram to illustrate the structure of a genome in a male somatic cell. So I was saying this again, it has been answered on what? This has been answered also on the channel, where we talked about the structure of what? The structure of genome in a male somatic cell. We talked about this to say that when you look at it, you need to see that we have got the XY chromosome. We need to have a title to say that we are saying this is supposed to have a male somatic genome, male somatic genome. So now, male somatic genome. So what we are saying is that we need to understand that this is what we are doing here. The caption, the male somatic genome is the caption, so we can just save our X and our Y chromosome. And then the male somatic genome, we are saying it is the, you need to know that we can draw this as to say that we have got a long X and a small x, which this one is represent the short is representing Y chromosome. This is representing the Y chromosome. I'm saying we know that a male is XY, but when we draw, we need to draw a diagram which is similar or which is like this one, where we have got the long and short chromosomes there, which are representing the Y chromosome, which is given by what? By that short Y chromosome that is represented by that X there. Explain why Henry's 80 and not his wife was responsible for the sex of the children. Here, what you need to understand is that question 2.33, why Henry was and not his wife was responsible. You are talking about this question. I am saying we also answered it, which you need to understand that the gender of a child is represented by what male give. We are saying in this case, you need to know that he is a male who gives an XY chromosome, and then he gives the XY chromosome, and his wife, his and wives give what? An XX. XX, we are saying that which gives an XX chromosome. So now XX will always give to what? To female or to a girl child. So now he must give we are saying this man, he must give a what? Mr. Henry 80, Mr. Henry, Mr. Henry 80 is to give the Y chromosome, which defines the gender of children. The gender of children is defined by his donation of the X, which we are saying that if he gives an XX, we have got a what? A baby girl, or if he's going to give a Y and Mrs. Wife is going to give and I mean the wife in that case they would have a what? A boy child. So this is a sex determination question class, that males are the ones who define the gender of the child of the children, because the X chromosome is XX defines the girl and XY defines a boy, where the Y must come from what? From the father. That is what we need to understand in this case. So we are given this question, which represents six chromosomes of males of equal length are referred to as ZZ. We are saying female birds have six chromosomes of an equal length and are referred to as ZW. ZW, we are saying it is what? Unequal, and it is representing what? It is representing female. And ZZ is referring equal, which represents the what? The male. So the inheritance of head feathers in sex linked is sex linked. Head feathers, this is sexy, this is sex linked. So what is happening is that and the gene for the gene for the gene for head feather color in birds is only found on the Z chromosome. It's only found on the Z, and the W chromosome does not contain. This one, we are saying it is sex-linked, hence W chromosome does not contain a copy of this gene. Red head feather R are dominant over the black feathers are recessive R are there. So what is given here is that the question is saying using the letters R to do a genetic cross between a heterozygous male a heterozygous male red-headed feathers and a female with black head feathers to show the phenotypic ratio. So we are given that the male head feather in color in birds is only found on the Z chromosome. It is only found on the Z chromosome. And then we are also given that we are also given that in all birds, the sex chromosome of males are of equal length and are referred to as ZZ. So now, what we need to look at is that female birds have sex chromosomes of an equal length that are referred to as ZW chromosomes. So we are given that to say that using R and this one do a genetic cross between a heterozygous male with red head feathers and a female. So we are saying the male is we are saying a heterozygous male which is heterozygous. The heterozygous female is supposed to be RR is crossed with what? Is crossed with the W chromosome does not contain the gene. So what we are saying is that we have got also this is a ZZ, and then we are saying it is crossed with ZW, but we have got what? We have got our black, which is recessive R. Our recessive R is this one. So we are saying phenotype P1, phenotype is equals to red head feather, red head feather is crossed with black, is crossed with a black feather head, black head feather. So having that, we have got our genotype. Our genotype, we are going to have our Z. We have got our Z, which we have got RR is crossed with what? With the Z, we can say it's crossed with RR. So now our meiosis, and then we have got our gametes. Our gametes, we are going to have R, and then we also have what? We also have R is crossed with what? With R, because it's meiosis, it gives us one R, and then we have fertilization. We have fertilization. Our fertilization is going to take dominant R crossing with that one. We have got RR. We have got this R. Recessive R is crossed with this R. We have got RR. So our genotype, we have got we have got that and that. So our phenotype is going to say one red head feather as to one black head feather. So we are saying the phenotypic ratio is going to give us one red head versus one black head. So you have done the genetic cross. The genetic cross is being done. Remember, we said that the allele is sex linked, and then it is carried by what? By Z. It's carried by Z, not by W. So that is it. 2.5, we are given in certain type of plants, the gene that determines the leaf shape and color of flowers are found on the same chromosomes. The leaf could be either it could be either wrinkled or it can be smooth, and the flowers could either be purple uh, or red. So the diagram represents the arrangement of alleles on the chromosomes in plant A and B, which we are given there. So now, chromosome plant A, we are given that this the leaf is what? This we can say is wrinkled leaf. This is a wrinkled leaf, and then also it's a wrinkled leaf versus what? Purple flowers. And purple flowers. This is because we have got a dominant E there and a dominant D there. So the other opposite one, the second plant B, it is actually smooth leaf. We have got a smooth leaf versus what? Versus smooth versus a what? A red. We are given red what? Red flower, which is because we are having all the recessive alleles that are there. So this is what we need to take note of, class. So identify the phenotype of plant A. The phenotype of plant A, it's already given red leaf and purple flowers. We are saying it is not red. We are saying it is wrinkled leaf with purple flowers. Wrinkled leaf, and purple flowers. Where are we getting this answer? We are getting it from what? We are getting it from the concept that this is a D, and then we are having also an E and E, which I'm saying that when we have that expression, when we have this expression, this represents what? Ringwood leaf color, and the ringwood leafy, and then the color is what? Is purple because of the dominant E that we have there. And then the genotype of plant, the genotype of plant B, the genotype of plant B, it is actually given here. We have got the genotype of plant B. We are taking this. We are taking the genotype of plant B. We have got our genes that are given here, and that one. So our genotype is going to be all recessive. We have recessive D and we recessive E, as it is given on the what? In the question there. is recessive. And then plant that has a chromosome with with homozygous. Identify plant that has a chromosome with heterozygous with homozygous alleles. Plant that has got chromosomes with homozygous alleles. Homozygous alleles. It is what plant B. Plant B has got homozygous alleles. We are saying it is plant. It is plant B. Plant B, we are saying this is homozygous because we have got this expression that we are given there to say this is plant B that we have, which is actually transferring to give us this answer that it has got homozygous, which we said that it means what? It means red and what? It means smooth and red flower. Both recessive alleles are given. 2.51, 2.5.2. Determine how many possible different genotypes gametes of plant A can have. Plant A possible genotypes. Determine how many possible genotypes of plant A can have. Genotypes possible. Determine how many possible different genotypes of plant A gametes of genotypes the gametes of plant A can have. So what we are given is that plant A, we are given that plant A, we are given that it is both heterozygous. Plant A, we are given that it is heterozygous. So we have got our plant A, it is equals to, we are saying it is D and D and then E and E. So our possible determine how many possible different genotypes the gametes of plant A can have, genotypes of this can have. So we are saying in this case, we can have, we are saying the genes are linked and we assume no crossing over. Plant A can only produce what? Two gametes, can actually produce two gametes genotypes, which is, uh, we can say that it can have, we can have possibly, uh, we can actually possibly have, I want to just show you something. We can have this D and this E, this D and this E, and then we are saying this is DE and then oro, and we also have this D and this E, and then we have got what? DE. So this is what we are having. So we are saying the certain type of plants that determine that, and the leaf could add the ring could and what. So we can have that and that. We can also have, we are saying we can also have, or we can also say that, let me just quickly show it clearly again, show it that we can also, we are saying this D, and we are saying this D, and that dominant E, we have got that one. So we can also have this D. We can also have this D and this E. We have DE, and then we can also have this D and small e. We can as well have we can also have small d combining with capital letter E. Then we can have ED. So I'm saying these are the possible the possible gametes that we can have. The possible gametes that we can have. So we can have 2, 3, and then 4, which we are given which are caused because we have got what? We have got this combination. These are the possible question 2.5.2. And then plant A and B are crossed with each other. What percentage of offsprings will have wrinkled leaf and purple flowers? Yes. Plant A is crossed with plant B. Plant A, we said that plant A, it is D, and then it is DE, and then we are saying it is crossed. This is 2.5.3. It is crossed with what? With DDEE. So now we are saying if they are crossed, we are saying what percentage can be what? What percentage? What percentage? What percentage can be? What percentage can be wrinkled? So when you give these guys, I'm saying the D hybrid cross, if you look at this, you work it out, you shall find out that we have got a 50% as to 50% chance of this. We have got a 50% to 50% chance. So what I'm just doing here is I'm giving you this. I'm saying it is also calculated, also refer to channel as well in terms of the dihybrid crosses. They are well explained, they are well worked out again, which we need to see that they've been proven, and you can be able to get good answers to these, because we are saying these answers, you can actually see that we are saying we have got a cross, which is cross in that regard. So therefore, what we need to know is that the other part, this one, we're saying it is what it is. This is, we are having a recessive expression that we have there. So in this case, we can have one gamete that can come out to this side. We have got one gamete that can come this side. One gamete is going to result here. We remain with one gamete, which is going to be what? DE. We have got one gamete that is going to be DE. So if you cross it with that other side, which we said that when we cross it, it can also give us what? We can also get that answer to find out that it will give us a 50. If we deduce it, the answer will come up to say 50 as to what? It can give us a 50-50 to say DE, or we can have DE here. So when we cross these, they can give you a 50% chance that you can get in that respect. So we are given that again, the Pukato was a large wild goat with long curved horns. For centuries, it lived high in the mountain range that divides France and Spain, which were given there. And then they were once abundant, but extensive hunting reduced their numbers to only 100 individuals in 1899. The last, known as the Celia, was found dead in January 2000 by park rangers, and then using the same techniques used to clone a dolly sheep. Researchers could transplant the DNA from the ear tissue into the ova taken from the domestic goats to create 430 embryos, of which 57 were implanted into surrogate airways. Only 7 of the embryos resulted in pregnancies. So we are saying when the implantation was done, when they were implanted, only 7 survived. So The question is saying, name the biotechnology process used to make the identical copy of the Picard. We are saying this is what? This is cloning. This is what? This is cloning. Cloning. And why was the tissue of the shape of the serial ear was used? We are saying, actually, we are saying the ear which was used is because it has got the favorable, it had the favorable traits. It has the, we are saying the desired traits. It has the desired traits. We are saying it was easy to sterilize, and also we are saying it was actually having the diploid cells. It has the diploid number of cells that were actually needed in this case. The diploid, actually full health diploid cells. It was the perfect donor, nuclear donor. It was the perfect nuclear donor that we can give in that respect. Explain the procedure used to create an exact copy of the Picasso using the tissue sample obtained from the ear. This question, what it wants you to explain, it wants you explain the reproductive cloning. So I'm saying reproductive cloning class, it is also referred to channel on reproductive cloning. It is actually done. Reproductive cloning referred to channel. It is actually done in this one. So I'm saying you need to understand that it is a step-by-step process, which we are saying that researchers must actually harvest the somatic. They actually harvested the somatic fibroblast from the cilious ENA and then cultured the cells, which we again need to understand that they removed the nucleus from the what? From the domestic goat. The nucleus from the domestic goat was removed, which we are saying again, micro-injected a bucard of fibroblast nucleus into the what? Into the enucleated egg, zipped the hybrid cell with what? With an electric pulse to actually fuse the membranes and actually initiate the cell division, cultured the resulting embryo to blastocyte stage, then transferred the 57 embryos into the surrogate goat or airways. 7 pregnancies resulted, which we are seeing from the channel, from the context there, but only one live birth, which died minutes later due to lung defects, whatever that is given in context. So the question wants you to explain the positions or the process of what reproductive cloning. We have got the surrogate. We have got the surrogate mother, and then we have got the donor mother, donor or champion mother. So remember that we have been talking about this to say that this process, you need to know that we have got an egg cell with a nucleus, and then we have got a diploid cell that is taken there, and then we said that the nucleus is removed, and then we have got here the nucleus which is taken, and then we say that the egg cell is taken, and then the nucleus inserted into the surrogate egg cell, into surrogate empty egg cell. You understand? So now we are saying when this is done, we are going to have it like that, and then we are saying it is stimulated to divide stimulated to divide through electricity. Remember, we said that it is jump-started division because of what? Because of an electric to divide by electric impulse. By electric impulse. And then what happens is that it is taken and then inserted into the surrogate. It is inserted into the surrogate mother, and then we are saying the surrogate mother will give birth to, we are saying it gives birth to a young one alike to the champion donor. This is basically what we are saying. This is basically what I have just described there. That's what I have just described with regards to what to this whole process. So the nucleus of the donor mother is removed in order to get rid of characteristics from what? From donor mother. From this characteristics of the surrogate. That is what is happening in this case class. That is what you need to understand. That is what you need to take note of. So the mother, the birth is going to be, the young one is going to be exactly that one which we are saying is going to be a replica of the champion. It is going to be a replica of the champion, which you can see there. So the diploid cell, we talked about the ear that was taken there, the ear nucleus, which was taken there. So this is just evolution. Oh, wait. Let's just quickly check again. Do we have? Okay. Describe how the researchers could have calculated. The next question is saying, describe how researchers could have calculated the percentage success rate of this one. Question 4, 1.4, 3.1.4. 3.1.4, we are saying number of pregnancies divided by the number implanted is what we're gonna do here. Question 3.4, describe how researcher could have 3.4, this one. How the researchers could have describe how the researchers could have done that one. Is just a simple question that we can quickly push together right now. What we need to understand is that this question, it wants you to say that the percentage success is equals to percentage success, we are saying is equals to, we are going to say number of pregnancies over number of pregnancies divided by the number implanted embryos divided by we are saying over number of implanted embryos over number of implanted embryos multiplied by 100 over 1. That is what the question wants you to understand here, which is equals to, if we look at this, we just say number of pregnancies 7 over implanted is equals to what? Is equals to 57, which we say multiply by 100, and then we can get our answer as what? Is we can give it through a calculator, 12 point what? 12.3%. And then we get our answer as 12.3%. You understand? That is the success. Number of pregnancies over number implanted times 100 of the number implanted embryos times 100. And then you get your answer there. The next question is saying the diagram shows possible relationships between hominids. There are possible relationships between hominids that we are given here. So what is it that we need to do? We are given this evolutionary diagram, evolutionary graph that we need to understand. And in this case, let's just answer the questions that are there. Identify the type of diagram shown. This is a cladogram. The graph that is shown is a clado. You can just say it's a cladogram. It's an evolutionary tree diagram. It's called an evolutionary tree diagram. How many genders are represented in the diagram? The genders that are represented in the diagram, we are looking at, we just count the labels, ne? We have got Ardipithecus, we can say Ardipithecus, we have got Australopithecus, all of them which are Australopithecus, we count them there. And then we have got also Paranthropus. We have got Paranthropus. And then we have got Paranthropus. And then we have got what? We have got Homo. So our answer is that how many genders are there? There are 4 because we are counting. We are saying Ardipithecus is 1, or Australopithecus, whatever Australopithecus, Africanus, and Afrensis, is the all 4 under 2. Paranthropus is another genre, and then we have got also the Homo. So our answer is that we count all of them. They are basically 4. And then name the species that is the oldest. The species that is the oldest, we are just seeing the one that existed first. The one that existed first is the oldest. So obviously, like in terms of people are born, the oldest one, it is called, the oldest one is called Ardipithecus Ardipithecus ramdas is the oldest. Ardipithecus, the oldest is Ardipithecus. Ardipithecus ramdas. And then the species that share the most recent common ancestor with Australopithecus africanus, that share the recent common ancestor with Australopithecus africanus. Australopithecus africanus, the Australopithecus africanus is this one. So we are saying the one that shares the common ancestor, the one that shows the we are saying the one that shares the most recent common ancestor with Australopithecus africanus. You shall see that when you look at that Australopithecus africanus, you shall see that we look on the graph. When you look on the graph, Australopithecus africanus, it is show it Australopithecus africanus. It shares a common ancestor. It shares a common ancestor. Australopithecus africanus. It shares a common ancestor uh, with we are saying it shares this one, the Australopithecus africanus and the Paranthropus. They are they are coming from the same, as you can see, they are coming from the same, from the same latest ancestor, which we are saying that they all come from what? They are all coming from Australopithecus afarensis. So it's Paranthropus robusta. Paranthropus robusta is what you are supposed to see. Paranthropus robusta is what is given in this case, and then name two forms of evidence that could have been used to support the information in the above diagram. Uh, Sources of evidence that can be used, we can talk about genetic evidence. Genetic evidence. Genetic evidence can be used. We can talk about fossils. We can talk about fossils. We can talk about embryology. We can talk about many other sources of evidence that we can talk about, and then give the name of the Homo species that used weapons and tools for the first time. The Homo that used the weapons, if you look at this one, we always know that the Homo habilis, if you can check also on the previous presentation, this has been said, describing how the discovery of Homo fossils support the out of Africa hypothesis. You find out that the out of Africa, we can find out that the oldest Homo fossils, they all exclusively come from what? Africa. They were exclusively discovered in Africa. All the oldest Homo species were discovered in Africa. Were discovered in Africa. They were discovered in Africa, and then we are saying those are the oldest. All the oldest fossils were discovered in Africa, and also younger ones, they appear in Europe and Asia. Younger ones in Europe and Asia. You understand? The younger ones were found in Europe and Asia, which actually shows that we have got a migratory origin from Africa. That is what we need to understand in this case. Explain the significance of three changes of this human skull that shows progression in evolution. We can talk about the significance. One, cranial capacity. Cranium capacity. Cranium capacity from which actually changed from, we are saying, from 400 to 1,400 cubic centimeters to 1,400 cubic centimeters, which actually enables in terms of thinking. It actually promoted critical thinking, And then we have got the prognatism shrinked. The prognatism decreased. The prognatism decreased, which resulted in a more flatter face that we actually talk about there. And then also detention decreased. It also decreased smaller teeth, smaller teeth, smaller teeth, smaller teeth, which also came, which actually showed that softer diets were eaten, showing eating and dependency, eating of softer diet. We also talk about even all these which we talk about the brain reflecting bigger brains. We say this reflected, it actually reflected bigger brains, and also the sophistication of the modern man that we can think about as we talk about evolution. It is a question that shows the investigation determining which type of inheritance accounted for genetic disorders in Boba males. All dogs have a chromosome count of 78. That is what was done, the procedures and everything. So results of the investigation are shown there. Results of the investigation are shown. So now, what are we supposed to look at? We are asked to look at identify the independent variable. I'm saying independent dependent variable questions class. They are always given an easiest question that we must mark and get right here. So an investigation was done, investigated determining which type of inheritance accounted for genetic disorders in accounted type of inheritance. We are saying type of inheritance accounted for genetic disorders in dopamines. So now, what is the independent variable? Independent variable, we are saying it is actually the type of the type of inheritance pattern, type of inheritance pattern, type of inheritance, type of inheritance pattern. It actually gives us it is the independent variable type of inheritance pattern, which we can say that it can be autosomal dominant or autosomal recessive, or can also just be saying sex linked. And then state 2 ways in which the scientists ensure the validity of the investigation. I say these questions class are always just there. We talk about big sample size, which is given in this case that they've got 1,200 dogs. 1,200 dogs. And then also we can talk about using a single breed of dogs. Using single breed of dogs. Using single breed of dogs, it actually removes the genetic problems that might arise. And then it also avoids other genomes to be added in this case. You understand? And then explain why there is no need to keep the job age constant in this type of investigation. Uh, the age, why there is no need to keep the age constant in this investigation. It is because inheritance patterns is actually genotypically affected, not influenced by the age of the dog. So age is simply irrelevant to allele classification. Age is irrelevant. Age is irrelevant. It's actually irrelevant to allele classification. Allele classification. So what we are saying is that uh, inherited patterns is actually genotypic. It is in the genes. Inherited patterns is genotypic. It is affected by genes, not by age. You understand? That is one thing that you need to understand. I explain why the inheritance of autosomal dominant disorder is so much higher than the inheritance of sex-linked disorder. Why is this like that? So what you need to understand is that what you need to understand in this case is that autosomal dominant disorders uh, is only because one mutant allele is needed. No, we are saying only one mutant allele is needed because only only one mutant allele is needed. Only one mutant allele is needed in this case. You understand? We are saying one mutant allele is needed. There is no carrier that can hide and seek, and also decades of selective breeding for short traits inadvertently propagated dominant disease alleles, whereas sex-linked disorders hide in healthy carrier females. So what we need to understand is that only one mutant allele is needed, no carrier hide. No carrier hide in this case, no carrier hide, and uh, plus decades of selective breeding, plus decades of selective breeding uh, to show traits. To show traits. So whereas sex linked basically sex linked disorders hide in healthy career females, hide in healthy career hide in healthy career females, which reducing their visible frequency. Reducing their visible frequency, to say we can see few of them because of that. We can see few of them because some of the sex-linked cannot be seen in the healthy females because they are not seen there. That is what is happening. In terms of the pie chart class, just go the pie chart. I've already worked it recently. It's recently worked. Just soon. Check on biotechnology, biotechnology, and genetics video. Biotechnology and genetics video. For those who are following Metric Hub, I'm sure that you will never go wrong because this question, it was worked last week, and not enough, it is coming again now. It is coming right now in this exam, which you can see that I'm telling you that if you can follow Metric Hub, you will never go wrong. We only need to understand that we need to convert information given into degrees. Uh, Calculate, it said, draw a pie chart to represent the information provided in the table. The information provided in the table. Otherwise, I was saying again, in simple terms, you might draw using percentages because here you know how big is 71%, how big is 11%, and each of them. So therefore, you can also draw using what? Using the percentages that are there. Or else, or else, or secondly, we are saying again, you can draw it by converting the percentages into degrees. You multiply, you do that, you do each of them 71%, 11%, 10 and 80 is 71, it's 11 out of 100 times 360 over 1, it is 10 over 100 multiplied by 360 over 1, it is 8 over 100 multiplied by 360 over 1. So you do this, you get all the various degrees that you should get on each of these. You get all the degrees. When you get those degrees, for example, you can see here you get 256 degrees. On that other one, we can work it. 11, we get 40 degrees. We get 40 degrees. On 10, we get 36. And then the last one, 8, we get 29. These are the degrees, and then you just go draw your pie chart, and then you know that the biggest one is 200 and what? 256 is the biggest one. 256 is big. Which is going to be drawn like that. I don't know. Let me see. I'm just saying that's how you draw your thing. So we are actually going to draw something like that, 256, and then we have got 40. 40 is like that. The concept is just showing the variation of the degrees that we have, and then we have got 36 and 29. We have got the smallest, which is 29, which is 36, which is 40, which is 256. And then you are giving your labels to say what is the meaning of each of those. Autosomal, autorecessive, sex-linked, you label each of them. you can label them. What is the meaning of all of these? You label them the way you label them. But the concept is for you to be able to know how do you draw these things. That's what is important. 3.4, uh, uh, use the example in the passage and explain how dogs initially may have evolved through natural selection from an extinct species. So what we need to do is here, this question, you are explaining natural selection in terms of the development of what uh, natural selection in development of dogs. So in other words, in this case, what we need to know is that natural selection is the process that leads to this. So an extinct gray wolf population scavenged human camp waste in maybe some places, and then mutants carrying extra amylase gene copies digested starch efficiently and thrived, reproduced, while the low amylase wolves starved. Over millions of years, directional selection took place and then enriched the amylase duplication allele, which resulting in these types of dogs into uh, domestic lineage dogs, which survived of starch-friendly home-cooked foods. So, in other words, you are just describing from the wolves how the dogs evolved from the wolves to become domesticated dogs, which is just a case of natural selection that we are talking about in terms of the understanding of natural selection that we talked about, which I've just narrated now, that there were that gray type of gray wolf population that also scavenged human camps, human camp waste rich in what in starch foods, and then mutant carrying extra amylase gene copies digested starch efficiently. So there is a concept of digestion of the starch. Other wolves could not digest and others digested, and then uh, they they digested it starch efficiently. They survived, reproduced, whereas those who did not manage to digest, whereas the other wolves starved, and over millions of years, selection pressure took over, resulting into uh, duplication of alleles. And those dogs or those wolves which survived on digestion starch more which we say produce more amylase, allowing dogs to benefit from what high starch diet of humans in a way, therefore survived and lived with humans. That is basically what you need to discuss there. Explain why breeds of domestic dogs belong to same species. Uh, we are saying because what is happening is because uh, they remain why all breeds of dogs belong to the same species. Here, what we need to understand is that they remained in one species. The reason is because of what? It's because they have got what 78 chromosomes. They remained because they have 78 chromosomes. 78 chromosomes, which is quoted in our essay here that they've got 78 chromosomes. There is we are saying they have got all dogs have got 78 chromosomes. So they have got 78 chromosomes interbreed freely. They manage to interbreed freely and produce fertile offsprings. continue to produce fertile puppies. They continue to produce fertile puppies. That is what we need to understand in a same continuous gene pool. That is what we need to understand here. The same continuous gene unit, the same continuous gene unity. That is the whole thing that we need to look at in this regard. So classy, that was so good and long discussion. Thank you very much for your time. And please just keep on following. I said for those who are following the channel, you will never go wrong. All the work is there. This metric examination preparatory that we wrote here now, almost 3/4 of the questions are directly done, not consider the concept, but the entire questions are well answered here. So please just keep following. For those who don't want to repeat, let's do this together. I'm telling you, work is well done here. All the work is here, and please don't keep on taking chances. Follow and let's do it together. Until we meet in the next videos, we are working towards revision. I saw that you want revision chapters or concepts on revision concepts on evolution. Everything we are going to revise it before the final examinations. Thank you very much for your time. Let's meet again in the next videos. Have a beautiful stay.